introduction of factors affecting enzymatic activity. We have learned what is an enzyme. Enzymes are also addressed as biocatalyst and organic catalyst. What are the difference between organic catalyst and inorganic catalyst? We have also learnt about mechanism of enzyme action. Two models of mechanism of enzyme action. What are they, ma? Lock and key hypothesis and induced fit model. Okay, induced fit model. And then we have looked into the action, activation energy and transitional energy. How enzyme substrate complex is formed, how enzyme substrate complex converts to enzyme product complex, and how products are being released. Till here we are done. Now factors affecting enzyme activity. There are three major factors which affects enzyme activity: temperature, pH and substrate concentration. Fine, right? We have learnt that there is something called as optimum temperature. Optimum temperature is the peak point at which there is maximum enzymatic activity. Okay, the enzyme activity is maximum. Enzyme activity is maximum at what point, ma? At optimum temperature. Enzyme activity is maximum at optimum temperature. Okay. As the temperature rises, there is an increase in rate of reaction, which means as the temperature increases, there is an increase in rate of reaction. There is an increase in the enzymatic activity. Optimum temperature, it is maximum. Once with decrease in temperature, it falls down again. With the complete rise in temperature, more than optimum temperature, if the temperature is rising out, that will destruct the structure of the enzyme. It leads to what? Denaturation of enzyme. This leads to denaturation of enzyme. Next is pH. Every enzyme's optimum pH will be decreased, will be specific to each enzyme. pH is specific to each enzyme. Okay. So here if you observe a different set of enzymes, their functions and their optimum pH. pH which is less than optimum pH or pH more than optimum pH, the enzyme activity will get reduced. At optimum pH only enzyme activity will be maximum, and that is specific to each enzyme. If you consider pepsin, okay, pepsin, the optimum pH is what, ma? 2.0. What is its role? Peptin will digest proteins in the stomach. Trypsin, what is its pH? 8.0. This digests protein inside the small intestine. Salivary amylase, you know, what is its main role, ma? To break the starch into maltose in the mouth. Its pH will be basic only, 6.8 to 7 pH. Lipase will digest lipids. Optimum pH will be 7.5 to 8. Very good. Maltase converts maltose to glucose. pH is 6.0 to 7.0. Renin, which is also known as chymosin, this coagulates milk protein, that is casein, in infants. Okay, whose optimum pH is 3.5 to 4.0. DNA polymerase catalyzes DNA replication, pH is 7.5 to 8. Catalase, this breaks down the hydrogen peroxide to water and oxygen, whose pH is 7. Okay. It is little difficult to remember the pH of all this, but have the basic idea like which enzymes will have basic pH, which enzymes will have neutral, which enzymes will have acidic pH. Those enzymes which are released in the gastric juice, in the stomach, their pH will be low. Those enzymes which are released in mouth, okay, and intestines, and especially in infants, those, in DNA polymerase inside a cell, okay, which will help in the synthesis of new DNA, those pH will be basic. Got it? Fine. And know the functions of each enzyme. These are the enzymes which are given in different chapters of your NCERT. Okay, if you observe DNA polymerase in mol bio and these enzymes in your human physiology, important. Though the digestion chapter is deleted, you to know the basic functions of these enzymes are important. Okay, clear with the two factors, ma? Temperature and pH. Now about substrate concentration. So first listen, substrate concentration, with the increase in substrate concentration there is an increase in rate of reaction. Firstly, with the increase in substrate concentration, rate of reaction increases. Once it reaches to the optimum, okay, unlike temperature and pH, what happens there? With the rise in temperature and rise in pH, the rate of reaction falls down, but here it comes to a saturation. Though adding up many substrate molecules, there is no increase in the enzymatic activity. For example, yesterday we have taken one example of sucrose, yes or no? I will take the same example and explain you. Sucrose is the substrate, and what is the enzyme? Sucrase. Sucrase is the enzyme. Just imagine this is a hypothetical view in a single cell, okay. There are 10 enzymes, 10 sucrase enzymes, let us suppose, okay. First you added up one substrate, that is sucrose, okay, then 2, then 3, 4, 5. So the rate of reaction will increase till 10, till you add 10 substrates, the rate of reaction will increase. After adding 10 substrates, though you dump 11, 12, 13, will there is any increase in the rate of reaction? No. There will not be any increase in the rate of reaction, because substrate and enzyme complex has come to a saturation. The enzyme capacity to hold the substrate has come to a saturation. Understood the meaning of saturation? Okay. Understood why there is no downfall or rise in the graph once it is reaching to the optimum point. Okay. And there is some phenomena called as Michaelis-Menten constant. What does it say? Km is the substrate concentration, okay, at which half of the maximum velocity is achieved. Okay. See, this is the complete Vmax, okay, complete velocity of the rate of reaction, Vmax, okay. Half of its concentration is achieved at Km, that is Michaelis-Menten constant, okay. Half of the velocity of the reaction, okay, half of the Vmax is achieved at what? Km. At some substrate concentration point, half of Vmax is achieved, and that point is considered as what, ma? Km, Michaelis-Menten constant. So Km is equal to Vmax by 2. Hardly there is not even a single question where they asked you on the application of this formula, but different statements and this formula is put on the statement based questions in your PYQs. Okay. You have to know what is Km, what is Vmax, what is Vmax by 2. What is Km, ma? The substrate concentration at which half of the maximum velocity achieved. Okay. Clear with 3 factors: temperature, pH, substrate concentration. Yes, ma.
Substrate concentration.
Okay. See, substrate concentration, I have explained you with one example. Whoever didn't got, listen once again, no problem, I am repeating it. For example, in a hypothetical view, in a single cell, you have 10 enzymes, okay. So you are giving out one by one substrate molecule to that cell, okay. There will be rise of reaction till you add 10 substrate molecules, yes or no? Till you add 10 substrate molecules, there will be rise of reaction. After 10 substrate molecules, though you add substrate molecules also, there will not be an increase in rate of reaction, because the enzymes have come to a saturation point with the substrate. There is no enzyme to catalyze the reaction even you add the more substrate. So that saturation point is known as the optimum point of substrate concentration. Once it has achieved here, though you increase the substrate concentration, it has come to a saturation level. Okay. This is the factor of substrate concentration. This is how substrate concentration is affecting the enzymatic activity. Second point you have to know is about Michaelis-Menten constant, which is known as Km. Okay. At this constant, what is achieved? Half of the Vmax is achieved. This is the maximum velocity, right? Half of Vmax is achieved at Km. Now you understood, everybody? Any doubts anywhere, ma? Yeah, so you are clear with factors affecting factors affecting the enzymatic activity. And now about inhibition of enzymatic activity. You are clear with this. Enzymes, substrates, enzyme plus substrate will form enzyme substrate complex. Enzyme substrate complex. This enzyme substrate complex in the presence of enzyme, that is at activation energy, turns out to be enzyme product complex, and this will release out the products. This is the normal rule. Now you have to inhibit this, okay, inhibit this process, that is known as inhibition. This happens in 3 ways, okay. First listen, what is competitive, non-competitive, allosteric. Competitive inhibition, the name itself is saying there is some competition. Competition between what? Inhibitor and substrate. You have observed, you have learnt that there are some active sites of this enzyme. Let us suppose these two are the active sites, okay. Inhibitor will mimic with the substrate structure. The structure of inhibitor is same as that of substrate, structure of inhibitor is same as that of substrate. So this will come and bind to the active site prior to the substrate. So it is affecting the substrate in binding with the enzyme, that is how it is inhibiting the enzymatic activity. Fine. Second is what? Non-competitive inhibition. What happens is, apart from active sites, whichever site is there is known as allosteric site. What happens is, inhibitor will come and bind to this site. This will change the active site of the enzyme. It will make the active site of the enzyme unfit for binding with the substrate. This is called as what? Non-competitive inhibition. Allosteric inhibition is also addressed as feedback inhibition. For example, we will consider glycolysis itself. What is glycolysis? Glucose, lysis, breakdown of glucose, okay. If pyruvate is abundantly present in the cell's cytoplasm, pyruvate is already present abundantly, will glycolysis will happen? No, because pyruvate is already present, the product is already present. So higher concentration of products will inhibit the rate of reaction. This is also known as what? Feedback inhibition. So you are clear on what is competitive inhibition, non-competitive inhibition, allosteric inhibition. Look at here, first binding site. In competitive inhibition, what happens, ma? Yeah, inhibitor will bind to the active site. Very good. Inhibitor binds to the active site of the enzyme. In non-competitive inhibition, this binds to the site other than active site, and it will make the active site unfit, so that substrate will not bind to the active site. Clear, right? And then binding site, allosteric or feedback inhibition. This binds to the regulatory or allosteric site, not to the active site. The products which are already present, this will bind to the allosteric site, so that the reaction will not happen. Clear till here? Second point, the products will bind to the site which is other than the active site. The sites which are other than active site on the enzyme are known as allosteric sites. Products are huge, so they will bind here and make the enzyme unfit for binding with the substrate. Okay. Second, structural similarity. Inhibitor is similar to substrate here. Inhibitor is not similar to substrate. There is no requirement. Okay. Here, product or inhibitor is one. Product itself acting as inhibitor, no ma. This may or may not resemble to the substrate. Effect on Vmax. Here the effect on Vmax is unchanged. Here velocity decreases, may increase or decrease. Quite a times questions are asked on the Vmax. How the inhibition is affecting Vmax in competitive, non-competitive, allosteric inhibition. Okay. And then effect on Km, substrate concentration, is increased in the competitive inhibition. Km remains unchanged in the non-competitive inhibition. This is variable again. It can increase or decrease. It may or may not affect in the allosteric inhibition. How Vmax and Km are affected in competitive, non-competitive, allosteric inhibition is important. Okay, these 2 points. These are anyhow logical points. If you understand what is competitive, non-competitive, allosteric, you will remember them. But these are the points to be remembered. Next is, can be reversible by substrate. Competitive inhibition is reversible, yes. It is irreversible, and this is also not easily reversible, okay, because it is completely regulating the entire mechanism. Yes or no? Product is regulating the entire mechanism. These are the key differences between competitive, non-competitive and allosteric inhibition. Just go through it for 2 minutes, and if you are having any doubt, ask me. Please take down. Hasib. Where did you see? Based on modulator type. See, modulator, regulator, which means that it is affecting on the mechanism pathways. So its effect is not static, okay, like how it is decreasing the Vmax, here it is remaining unchanged, here it is increasing the Kmax, apparent Km increases. Here it is not like that, it is modulating, regulating. Understood the meaning? Because it is acting on the mechanism pathway, biosynthetic pathways, okay. Done? Yeah. Look at it. Here what is important is these 2 points, that's it. Ignore this, not required. Example here, malonate inhibits succinic dehydrogenase, okay, from succinate to fumarate. The enzyme which is involved is succinate dehydrogenase. So what is that which is mimicking the structure of succinate? Malonate is mimicking the structure of succinate. This you will observe in TCA cycle or Krebs cycle, okay. Here cyanide will inhibit cytochrome oxidase. Cytochrome oxidase is complex 4 in your ETS, okay. This is complex 4 in ETS, okay. This activity is inhibited by what? Cyanide. This is non-competitive inhibition, this is competitive inhibition. Here glycolysis, from the example of glycolysis, ATP inhibits phosphofructokinase, isoleucine inhibits threonine deaminase. You don't remember this, not a problem, but this you can easily remember in glycolysis pathway. Third step, where phosphofructokinase will catalyze the reaction. Phosphofructokinase, from fructose 6 phosphate to fructose 1,6 bisphosphate. Okay. So, at this third step what happens? ATP will be released, right? When ATP is huge in the cytoplasm, it will not allow the function of phosphofructokinase. ATP is already present, so it will not allow the function of phosphofructokinase. Understood? Done. Enzyme inhibitor complex, here it is formed. Here it can bind both to a free enzyme or enzyme substrate complex. Okay. Even after the formation of enzyme substrate complex also, it is binding to the allosteric side and making it inactive. That can also happen. And to single enzyme also, inhibitor can bind other than the active site and make it unfunctional, unfit for substrate to bind. Two things, either of the things can happen. Here in allosteric inhibition, enzyme inhibitor complex will be formed, but usually this induces what? Conformational change of the enzyme. These 2 points, that is it. Here example is taken from TCA cycle ma. Here it is complex 4 of your electron transport system. Here it is third step of glycolysis. Questions can be asked. Okay, because all those 3 are there in your NCERT, right? TCA cycle is there, complex 4 ETS is there, glycolysis is also there. Done. Now, classification of enzymes. Okay. It is the International Union of Biochemistry which has come forward to classify the enzymes based on class, subclasses and all. So, here at this level, if we are clear with the classes, it is enough. Okay. So, first enzyme class, if you consider, we have 6 classes. First is what? Oxidoreductases or dehydrogenases. What enzymes are put under this class? Those enzymes which catalyze oxidation reduction reactions. Okay. When oxidation reduction reaction happens, what happens ma? There will be a transfer of electrons or protons. In those reactions where oxidation reduction reaction happen and there is a transfer of electrons and protons, such enzymes are kept under what? Oxidoreductases or dehydrogenases. Examples include dehydrogenase, oxidase, reductase, catalase, peroxidase. Remember the examples, very important, and the function of the enzyme also. Okay. This you can get in match the following, this you can get statement based. Second type of enzyme is what? Transferases. This will catalyze the transfer of specific functional group. Any specific functional group, if it is transferred, any enzyme is catalyzing that reaction, that is kept under transferases. Examples include transaminase, kinases. Okay, kinases will help in transfer of what? Phosphate group. Next is hydrolases. This will catalyze the breakdown of large molecules into smaller ones. Okay, using water. Examples include protease, lipase, maltase, nuclease. Lyases. This will catalyze the cleavage of the substrate into 2 parts using water, and this forms a double bond at the cleavage site. Example, aldolase, which is 5th step of your glycolysis. Okay, where fructose 1,6 bisphosphate, which is 6 carbon, is cleaved to what? 3 carbons, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Decarboxylase and carbonic anhydrase. Next is isomerases. This will catalyze rearrangement of molecular structures to form isomerases. Example include isomerase. Next is ligases. This will catalyze the joining of two molecules utilizing the energy ATP, and this forms covalent bonds between the molecules. Liases will involve in cleavage and ligases in ligation, that is joining. Okay. Example includes RUBP carboxylase, ligase and phosphoenol pyruvate. Okay. Take down the examples and all the 6 classes. The function of each enzyme, just note down in 1 or 2 points. Make it fast within 3 to 4 minutes. Ma. Asif, where is your running notes? Write it in running notes. Done right everyone? Now about cofactors. Okay, enzyme is functionally active when it is binded to cofactor. Enzyme will have two parts, right, which is protein part, which is addressed as apoenzyme, which is a proteinaceous part of the enzyme, and non-protein part is cofactor. Together, put together is observed as what ma? Holoenzyme. Okay, so here non-protein constituents are called as cofactors. They are bound to the enzyme to make the enzyme catalytically active. Three kinds of cofactors are identified. What are they? Prosthetic groups. Protein portion of any enzyme is? Okay, first you observe first example of prosthetic group, that is peroxidases and catalases. They will break down hydrogen peroxide to water and oxygen. Second example, heme is the prosthetic group and it is part of the active site of the enzyme. Two examples, peroxidase and catalases. Second is heme group. Clear? These are the examples under what? Prosthetic groups. Second is coenzymes. Coenzymes are nothing but the organic compounds. Let's say example NAD and NADP. Nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate. Their association with enzyme is not that strong like prosthetic group or a metal ion. It is transient or loosely arranged or loosely binded. Usually occurs during the course of catalysis. Most of the components of coenzymes are what? Vitamins. Coenzymes serve as what? Cofactors in number of different enzyme catalyzed reactions, and these are the examples. What you have to remember with coenzyme? Coenzyme association with enzyme is transient. It is not that strong like prosthetic group or metal ions. Second important point is coenzymes are mostly organic compounds like NAD and NADP. Okay, and vitamins. Okay, this is the association of coenzyme. Second and last segment of the chapter is metal ion cofactor. Metal ions are arranged by coordination bonds to the enzymes. How they are binded to the enzymes? By coordination bonds. Number of enzymes require metal ions for their activity, which form coordinated bonds with the side chains at the active site. At the same time, they form one or more coordination bonds with the substrate as well. Okay, this metal ion cofactor will bond to the enzyme via coordination bonds. Same coordination bonds will be binding with the substrate as well. Examples include zinc is the cofactor for proteolytic enzyme carboxypeptidases. Here what you have to remember? Specially examples. For prosthetic group, peroxidase and catalases. For hydrogen peroxide, and heme is the prosthetic group for the enzyme. Next is coenzymes NAD, NADP and vitamins. And then metal ions binded by coordination factors. Example includes zinc. Okay, zinc for the enzyme carboxypeptidase. Clear? Now we will move on doing the questions. Clear with the chapter, or you need a repetition of 10 minutes? No, no? Yeah. Once the question practice is done, we will move on with photosynthesis in higher plants now. See, we have 3 rows. 3 rows. 1, 2 and 3. Okay. Give me a paper and pen. Question to first row, this row. Just a second. What is the answer? 4. Yeah. Lipase ester bond. Nuclease, protease, amylase will work on. What is the answer? 4. Good. Next question to row B. Only this row. Option B. GLUT4 is what? Insulin, trypsin, collagen. Right answer is 2. Very good. This is NEET PYQ. Next question to row B. Even you can answer. Yeah. Who said? Okay. Just now we have learnt no, about carboxy peptide. Question asked in 2024. Next, fourth. Who answered? Question 2 here, ma. Inhibition of succinic dehydrogenase enzyme by malonate is competitive inhibition. Someone said 2? You said 4 only? Okay. Next. First you tell question to only this group. Lecithin, a small molecular weight organic compound found in the living tissue, is an example of? Very good. Question to this team. You can answer, take time and answer. Easy only. Options are not given. You have to tell the answer. Sequence after sequence. Done? You have to say the sequential steps. Arrange the statements in sequence. Wait, question to this group, no, only they have to answer. E, A, D, B, C is your answer. C, B, okay. C, B. That's it, no? Okay, we'll see. Substrate first binds to the active site of the enzyme. Okay. And then substrate enzyme complex is formed. Okay. Enzyme and then enzyme substrate complex is formed. And then you said D. Chemical bonds of substrate are broken. Wait. Chemical bonds of the substrate are broken and then they will release the products and then enzymes are free to bind to the another substrate. Correct only. Anybody objecting this sequence? Then say the explanation. See, from the enzyme substrate complex to enzyme product complex he didn't say. He is just mentioning chemical bonds of the substrate are broken. They broke only, no, they will form enzyme product complex. Okay, that point is not explained. He just said chemical bonds of the substrate are broken, followed by release of products, followed by, now the enzyme will be free. Free enzyme will bind with another substrate. Good. Lavanya, right? Good. Next. To this team, nobody should answer. Shush. They didn't answer, right? Okay. Okay, wait. Protein? Unsaturated fatty acids? Nucleic acid? Polysaccharide? Which option? A. Point to this thing. Next. Good. This question, this team. Silent and you have to answer only at that time. Can you observe this? Hold on a point. Can you observe this? Most of the questions are asked from the tables of this chapter. Huh? Terpenoids? Cool, relax. You are saying B, right? Terpenoids? Lectins? Alkaloids? Toxins? Answer is? Next. Wait, hold on. Question for this team. Huh? Options are not given, no? This is the question, right? Which of the following is not a secondary metabolite? We can say this. Lecithin. Point to this team. Next. 11th question. Wait. Afrin, did you say? You should be louder. Raise your hand and say next time, okay? Okay. Here, this question. Cellulose does not form blue color with iodine because it doesn't contain. Next question. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of. Tell me with what substrate it is competing? Succinate or succinic acid. Okay. This question. You have to raise your hand and yes. First option. Okay. Answer is already given. Adenine is? Okay. Anthocyanin? Chitin? Codeine? That's it. Next question. Primary proteins are also called polypeptides because third option, they are polymers of peptide monomers. Next, given below are two statements, statement based question. Look into it and answer. There should not be any sound. Yes, Afrin? Fourth option, good. Amino acids have a property of ionizable nature of amino and carboxyl groups. Hence, they have different structures at different pH. The question someone has bought me today. Amino acids can exist as bitter ionic form at acidic and basic pH. No, only at neutral pH, right? Only at neutral pH, which is known as isoelectric point. So the right answer is 4. Clear? Next question. In the enzyme which catalyzes the breakdown of hydrogen peroxide to water and oxygen, the prosthetic group is heme. Good. This will be the groups for what? Coenzymes, and this is for metal ion. Good. 17th question. Which of the following are not secondary metabolites in plants? They are primary metabolites. Okay, so you have to be silent, okay, and whoever knows the answer, raise your hand. Okay, Jyotika, fine, right? Okay. Next, based on the demand of audience. 2021 lo ochindi question. Repeated, huh? Okay. Whoever is giving quick answers, negative mark. Wait. 2 minutes, wait. Huh? Option A? Cool. Hold on. Identify the incorrect pair. Toxins are resin and abrin, not drugs. Drugs examples include alkaloids. No. Codeine and morphine. Okay. Alkaloids are drugs, vinblastine and curcumin. Lectins, concanvaline. Alkaloids, codeine, morphine. Toxins, abrin and resin. So the incorrect will be second, 20th question. I can't hear you. Third option. Here third option. Okay. Let everybody do. Anybody are having doubts with this question? Ask me. No? But listen. Adenine makes how much percent of DNA molecule? What will be the percentage of thymine, guanine, cytosine? From Chargaff's rule, it is clear that in a DNA or RNA, the percentage of purines will be equal to the percentage of pyrimidines. So, adenine will be equal to percentage of purines are equal to pyrimidines. Adenine, guanine, adenine, percentage of adenine will be equal to thymine, which is 30%. If you consider the whole as 100%, 60% is occupied, rest is 40. 20% and cytosine 20%. What is the right answer? Thymine is 30%, guanine, cytosine 20%. Got it? Any doubts? Understood? Amount of adenine is equal to thymine. Okay. Put together, purines are 50%, pyrimidines are 50%. Got it? So adenine is 30, so thymine is also 30 because it binds with thymine, right, in a double stranded DNA. And then guanine, the amount of guanine will be equal to cytosine. 20% guanine is equal to 20% cytosine. Clear with all the questions? Okay. So this is the level of questionnaire that is being asked from the chapter biomolecules. We are okay with the additional content also. We have connected the dots with the examples also. Okay. From enzymes, we have connected all the dots, including inhibition, classifications, okay, and enzymes, okay, which are mentioned in the complete NCERT. From the beginning to the end, we have completed everything. Once let us recall quickly what are the concepts we have learnt here. Starting from elemental analysis, followed by chemical analysis, then about acid soluble pool, acid insoluble pool, learnt about primary metabolites and secondary metabolites, then about carbohydrates, its classification, proteins, what are the different types of amino acids, levels of proteins, forms of proteins, primary, secondary, tertiary, quaternary structures, then about lipids, 3 different types of lipids we have learnt, simple fats, derived lipids, conjugated or compound lipids. And then we have also exercised the molecules, biomacromolecules given in the NCERT, structures of that. And then from the nucleic acids, we have learnt nucleic acid is a polynucleotide chain, how a single nucleotide is formed, how a single stranded nucleic acid is formed, double stranded is formed, what are the bonds involved, what is Chargaff's rule, what are the points of double helical model of DNA. And we have also learnt about different types of RNA. Then coming to enzymes, we have learnt the clear differences between organic catalysts and inorganic catalysts, and then we have learned the factors affecting the enzymatic activity, what is the action, mechanism of enzyme action, 2 models of mechanism of enzyme action, and then how temperature, pH and substrate concentration is affecting the enzymatic activity, what is the classification of the enzymes, okay. And then at last we have also learned about cofactors. 3 types of cofactors, prosthetic group, coenzyme and metallic ions with examples. That's it. Now the entire chapter is in your mind, right? At least a framework of this. Your revision also should go in this flow. So complete cell biology we have completed. Cell the unit of life, cell cycle, cell division and biomolecules. Now we are moving on to the chapter plant physiology. Just give me 2 minutes to down the PPT. Meanwhile, just go through this, practice the questions just for 2 minutes, ma. Here, you want water? Here everybody, sit straight. From your NCERT 11, okay, major set of questions are covered from cell biology and plant physiology. Cell biology is done, now plant physiology. From these 3 chapters, photosynthesis in higher plants, respiration in plants and plant growth and development. In photosynthesis in higher plants, what are the contents you have to go through? First is about discoveries. Followed by discoveries, you have to move on to action spectra, absorption spectra. Then chloroplast organelle. Inside chloroplast organelle, what are the photosynthetic pigments that are present? Followed by this, you have to know about cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. Followed by chemiosmotic hypothesis. Chemiosmotic hypothesis. Okay, and then followed by biosynthetic phase, which includes dark reactions or light independent reactions, where you learn about C3 cycle, C4 cycle, CAM cycle, including its bioenergetics. Okay, and there is one important concept, that is photorespiration, which is also known as C2 cycle. Last concept of this chapter, that is factors affecting photosynthesis. Factors affecting photosynthesis. What is the maximum level of questionnaire that you will get? 2 questions maximum from this chapter for your final NEET. Okay, done? You are clear with the concepts that we are going to learn? Okay. First, early discoveries. I don't want to waste much time for these discoveries. Okay, I will tell you by giving the standard equation of photosynthesis. What is the standard equation of photosynthesis, ma? Plus 12 H2O in the presence of what? Sunlight, chlorophyll pigment, which is present in the chloroplast. It forms the primary product C6 plus, and what is released as byproduct? This is the standard equation of photosynthesis, balanced equation of photosynthesis. From this only we will learn the discoveries also. Firstly, tell me, who is the scientist who said that carbon dioxide is essential for the process of photosynthesis? It is from Mole's half-leaf experiment. Mole's half-leaf experiment. Okay, and who is the scientist who said that water, sunlight are essential for photosynthesis and oxygen is evolved from the process of photosynthesis? This is by Jan Ingenhousz experiment. Jan Ingenhousz hydrilla experiment. How did Mole perform this experiment? Mole's half-life experiment. Can anybody tell? What happens is, a potted plant is taken which is placed in sunlight. Half of the leaf of the plant is inserted into a beaker which is having KOH solution. KOH will absorb whichever carbon dioxide is present in that half leaf. That plant is set under sunlight, placing half of the leaf in KOH for around 1 to 2 hours, and then the leaf is plugged, tested for starch. Half leaf which is exposed to sunlight prepared sugars and that showed blue color. Half leaf which is into the KOH solution showed pale color, indicating the absence of glucose, indicating the absence of photosynthesis. That is how it is said that carbon dioxide is essential for the process of photosynthesis. Jan Ingenhousz experiment, what did he perform? So this is Mole's half-leaf experiment. Yeah. What happened is, listen carefully beta, a beaker containing water is taken, okay, in that a testil funnel, can you see the testil funnel which is like this? In that a hydrilla plant is placed. That testil funnel is inverted and kept into a beaker containing water. Over this testil funnel, this hole is open, right? This is closed with a test tube. 2 such setups were made. One is kept under sunlight, one in dark room. After certain period of time, what is observed, you know? In the beaker that is kept under sunlight, bubbles are formed. Bubbles are formed. When these bubbles are tested, what gas it has? It is proved that oxygen is released. Okay? In the setup which is kept in dark, there is no reaction shown. With this, it is proved that sunlight and water are essential. Oxygen is evaporated or released from the process of photosynthesis. Understood 2 experiments? Now, glucose is the product of photosynthesis is said by von Sachs. This is the primary product of photosynthesis. Okay? When it comes to oxygen, oxygen discovery, who discovered oxygen? Yeah, Joseph Priestley. Who said that oxygen is evolved from photosynthesis? Very good. Okay. Till here it is clear, no? Mole's half-leaf experiment, Jan Ingenhousz experiment, that is hydrilla experiment. Okay. and oxygen discovery experiment, that is bell jar experiment. I think there is no requirement of repetition. Yeah, these are the points. If they are asking you who discovered oxygen, you have to go with the scientist. If they are asking you who said that oxygen is evolved from photosynthesis, you have to go with Jan Ingenhousz. Now, two more points. One is sunlight is required for the process of photosynthesis is said by this scientist. But what spectrum of sunlight is actually involved in photosynthesis is said by one more scientist, that is Engelmann. Okay? Engelmann's action spectra and absorption spectra. Two more scientists are left, Engelmann's experiment and C.B. Van Al. First we will learn this. Okay? Yeah. So what happened is, a unilateral sunlight is placed to a prism. Okay? Over a prism, what is placed? Unilateral sunlight. Once the prism, from the prism a unilateral sunlight is passed, from here it will be scattered to VIBGYOR, to 7 colors. Okay? VIBGYOR. Okay? To this exposed light, a green algae is placed. Okay? Green algae, which is Cladophora. What type of photosynthesis it conducts? Oxygenic photosynthesis. Oxygenic photosynthesis means at the end of photosynthesis, it will evolve oxygen as byproduct. So when oxygen is being evolved, to trace the amount of oxygen, what is placed over this Cladophora is aerobic bacteria. You have understood the setup. First, unilateral light placed over the prism, then it is scattered to VIBGYOR. What is exposed to this? Cladophora. Cladophora conducts what type of photosynthesis? Oxygenic photosynthesis. Over this Cladophora, what is placed? Aerobic bacteria. When this setup is being observed for 4 to 5 hours of time, what happens is, they observed that the Cladophora has been accumulated. It has formed two peaks, one at blue region and the other at red region. Blue region and red region, they have observed that aerobic bacteria got accumulated. Aerobic bacteria requires oxygen. They will get accumulated wherever there is more release of oxygen. Okay? So, it is proved that there are two spectrum at which maximum amount of photosynthesis conducted. One at blue region and red region of light, maximum photosynthesis is being conducted. With this, he has derived out two spectra, action spectra and absorption spectra. Okay? We will learn action spectra and absorption spectra with different pigments. Okay? Now, one more scientist, that is C.B. Van Al. He has conducted bacterial photosynthesis. Bacterial photosynthesis, where he said that the electron donor is H2S. If H2S is the electron donor, what has been released at the product? Only sulfur molecules. In that case, in oxygenic photosynthesis, when H2O is the electron donor, what is evolved? Oxygen. From this, he has derived a point that H2S is the electron donor for bacterial photosynthesis or anoxygenic photosynthesis. Water is the electron donor for oxygenic photosynthesis. So, here oxygen is evolved from water, not from carbon dioxide. Oxygen evolved from photosynthesis said by Jan Ingenhousz. It is coming from water, electron donor is water, as said by C.B. Van Al. Have you understood the specificity of statement? Discovery, Joseph Priestley. Oxygen discovery by Joseph Priestley. It is evolved from photosynthesis, Jan Ingenhousz. C.B. Van, till then all scientists believed that the oxygen is coming from carbon dioxide, but not it is from water is proved by C.B. Van Neel. How? By conducting bacterial photosynthesis. Bacterial photosynthesis or anoxygenic photosynthesis, where electron donor is H2S. Sulfur is released as a byproduct here. When water is the electron donor, what will be received? Oxygen. As simple as it is. Okay, they have performed very simple experiments, not complicated experiments. Okay, 10th class experiments. So see, oxygen is evolved from water. Water is the electron donor. Clear? Any doubts anywhere? This is the complete discovery part. So first, who said that carbon dioxide is essential? Mole's half-leaf experiment. Who said that water and sunlight are essential and oxygen is evolved from photosynthesis? Who discovered oxygen? Priestley. Joseph Priestley. Who said that the primary product is glucose? Who gave us action spectra, absorption spectra? Engelmann's experiment. Engelmann's experiment. Who said that? Who conducted bacterial photosynthesis? Who said that H2S is the electron donor for bacterial photosynthesis? Who said that water is electron donor? Same sign. This is the complete discoveries part now. So pigments, action spectra, absorption spectra, from here we will deal in the next class. Okay. Right away what you have to do is, you have to work on the biomolecules objective, fine, and photosynthesis discoveries part. Up till here you just work out. Tomorrow we will end up the complete class of photosynthesis. Okay. I'll keep